Welcome back, students. Welcome to Priya's Life Science Academy. This is your educator Priya. Welcome to the next class of Lac Operon. In the previous class, we have discussed about the basic points of Lac Operon, that at which point Lac Operon will be turned off, at which condition Lac Operon will be turned on. So now we will discuss a very important point of Lac Operon. I hope you remember the previous class where I have told you about the function of the three genes of Lac Operon, that is beta-galactosidase helps to break down the lactose into galactose and glucose, and I told you about the enzyme permease, that the function of permease is to make the entry of the lactose inside the cell. Permease creates a pore in the cell membrane, and by the help of that, lactose can enter inside the cell. Okay, and then lactose can turn on the Lac Operon by inhibiting the Lac repressor, which I told you. But point to be noted that there's a very interesting point you should remember, that I am telling you that permease is needed for the entry of the lactose, and at the same time I'm also saying that lactose is needed for the synthesis of permease. If lactose doesn't enter inside the cell, then it can't inhibit the Lac repressor from binding to the operator, and then the enzymes can't be made. So two contrasting points are here. Permease is needed for the entry of the lactose. Lactose is needed. If lactose is not there, how permease can be made? The answer resides here. There's a point known as the leaky expression of the Lac Operon. Now, what is leaky expression of the Lac Operon? The repressor, they just not only bind and stay, they bind and dissociate. And in the split second, after the one repressor unwinds and before another binds, in that second, the RNA polymerase can bind on the promoter and transcription can be initiated. I am showing you by the diagram that this is our operon. This is the promoter region, this is the operator region, and these are the three structural genes. I hope you remember Z, Y and X. So in the promoter region we know Lac repressor binds. Okay, this is Lac repressor. But what is the case? These repressor, they just do not bind and stay, they dissociate also. And when they dissociate, at that time there is no Lac repressor present. So at that time the operator is free, and before another repressor can bind, the operator region is free from the repressor, then the RNA polymerase can bind easily and can start the transcription. Then the initial enzymes are made, then beta-galactoside is produced, permease is produced, and trans-acetylase is also produced. Okay, so this permease is very much important for the initial transportation of the lactose inside the cell. So here in this case, even in the absence of the inducer, the Lac Operon is turned on because permease is made, and then this permease can allow the lactose to enter inside the cell. Okay, because we know that permease allows the entry of lactose inside the cell. So suppose this is the bacterial cell membrane, and lactose can be incorporated only when permease is made. Okay, and this is known as the leaky expression, and this is very much important because you may wonder that ma'am is saying that permease is needed for lactose to entry, and again I'm saying that without lactose permease can't be made. But how the first lactose is incorporated? Can you understand the point that permease is needed for entry of the lactose, and I'm saying if lactose, the main inducer, is not present, the permease can't be made. Okay, so how this is happening? This is happening, and this answer is hidden in the split second. And when one repressor dissociates and another comes, in the split second, the frame is free, then the RNA polymerase binds and start the transcription, then the initial permease is made, and that initial permease is allowing the entry of the lactose. And then lactose can turn on the Lac Operon, and then if glucose is not present, the initial permease is made, so these initial permease can make the entry of the lactose inside the cell. So even glucose is present, there is always a low level of transcription in case of Lac Operon. Always low level of permease is present in the cell. That is why when there is absence of glucose, there is still little permease present which can make entry of the lactose inside the cell. Okay. So this is the reason behind this. Now we will read some operon mutations. Okay, there are some mutated form of the genes which are present on the operon. So we have read about a lot of gene present on the Lac Operon. Firstly, we will see what are the mutations present in lacI gene. I told you I is for inhibitor that produces Lac repressor. Okay, the gene I is inhibitor and which produces Lac repressor. So I for here inhibitor which produces Lac repressor. So there are several forms of mutations in the I gene. Okay, the functional form of lacI is lacI plus, which produces functional form and or wild type form of the Lac repressor, which is wild type. Okay, but if there is I minus mutation, if there is any kind of mutation in the inhibitor gene, then one kind of mutation is lacI minus, which produces non-functional repressor protein, okay, which can't function. Number 2 is IS. IS is known as super repressor. Okay, it can bind to the operator but cannot recognize the inducer allolactose. As the name suggests, the super repressor is a massive form. Okay, even if the inducer is present, that is lactose, the super repressor can't be omitted. Like in normal case we have seen when our inducer molecule, that is lactose, is present, that can block the inhibitor, that is Lac repressor. But whenever there is IS mutation, which is a super repressor, super repressors can't be inhibited by any kind of inducer, that is lactose. Okay, we will come to this later by giving you example. Now another mutation of I, that is I minus D, that is dominance, that is known as dominance mutation. I minus D produces a very fragile kind of repressors which cannot work properly. We will come to this later. And so this is all about I. So the gene I for inhibitor, the wild type form is I plus, which produces the wild type form of the Lac repressor, which can be blocked by the inducer, that is allolactose or lactose, and there is an that is wild type. The mutated forms are I minus, which produces non-functional repressor, so that means which cannot block the operator region. Number 2 IS, which is super repressor. These super repressors can't be blocked by the allolactose or lactose inducer, and I-D, they produces very fragile type of repressors which cannot which cannot block the transcription of the Lacoperon. Okay, so this is all about lacI. Now coming to the next mutation, that is operator mutation. We have seen that there is an operator in the operon frame. So this is the area for lacI gene, this is for promoter, this is for operator, and these are for three structural genes Z, Y and X, and here it's a promoter of lacI gene. Okay, so we have seen about the lacI mutations. Now this is the operator mutation now which we have to consider. Operator mutation, so the wild type operator is O plus, this is the wild type operator. OC is a mutation of the operator which is known as cis-dominant in nature. Now I will come to this point. There are two type of nature of the mutation. The one is trans-dominant and another is cis-dominant. Okay, OC is cis-dominant in nature, but the mutations of the inhibitor, these are trans-dominant in nature. Now let's look into what is trans-dominant and what is cis-dominant. Okay, we will look into trans and cis-dominant mutation. What is trans-dominant mutation and what is cis-dominant mutation? Okay. Trans-dominant mutation is seen in case of lacI gene. Okay, and cis-dominant mutation is seen in operator region, promoter region. Okay, the basic and the point is that trans-dominant mutation, the product of the trans-dominant mutation can travel, can transport and affect another reading frame from which it is produced. So, for example, lacI is produced from one reading frame and their product can travel and can affect other reading frame residing in other region. Okay. Other reading frame can be affected. Okay, but the mutation which are on the operator and promoter region, they do not produce any soluble product. They do not produce any soluble product. So their mutation is reside. They can only affect the mutation from which they are produced. For example, if the operator OC is here, it can only affect this reading frame. Their product can't travel to other region and can't affect other reading frame. Okay, so this is the basic rule of cis and trans-dominant mutation. From trans-dominant mutation, the products of the trans-dominant mutation can transport to other region and can affect other reading frames. But cis-dominant mutation do not, there is no such production of any soluble product. But so as because their products can't travel, they can only affect the reading frame in which they are produced. Okay, so operator mutation we have completed. lacI mutation, operator mutation and promoter mutation is very simple. P plus is the wild type and P minus is the mutated one. If promoter is mutated, then the RNA polymerase can't recognize the promoter and transcription will be halted. So this is all about the mutation. Okay, now we will see a very important point of the bacteria. These why we are telling about this cis-dominant and trans-dominant things, the answer is hidden in this. As because these bacteria are partial diploids or merozygotes. What is partial diploid or merozygote? I'm giving you an idea. This is bacterial cell, and you know bacterial cell contain two chromosomes. One is the plasmid Another is the bacterial own chromosome. This is the plasmid DNA and this is the bacterial chromosome. Okay. Plasmid DNA contains lacI gene. This chromosomal DNA also contains lacI gene. Plasmid DNA contains operon. This chromosomal DNA also contains operon. So, this F plasmid contains some of the chromosomal gene in themselves. That is why they are known as partial diploids. They are not fully diploids because not all of the chromosomal gene is present in the plasmid. Some of the chromosomal genes are present in the plasmid. Among them, lacI and operon is present in plasmid as well, along with the chromosomal DNA. That is why they are known as partial diploids. And because of this partial diploid system, we have to consider two reading frames. And that is why our concepts are coming of cis-dominant and trans-dominant. Now, as we have understood our two basic things here before the class, that is, we have understood the different kinds of mutation in the Lac Operon which are happening. We have understood what is merozygotes or partial diploids. Now we can easily understand the different situations. Now here we will understand the different situation of the Lac Operon. Okay. Now first now we will understand the different conditions at which Lac Operon can operate. There are lots of conditions because of the mutation present, like which mutation can produce which type of expression, when there is absence of glucose, what will happen, when there is a presence of lactose, what will happen. So by keeping all of the mutations in combination and by the presence of lactose and absence of lactose, we will see what will be the outcome of the Lac Operon. So these are some examples you have to thoroughly understand. I'll give you certain examples in this class, and in the next class we will discuss some problems, which will be asked in your exams. Okay, so first of all we will understand the lacOC mutation. OC mutation, as I have told you, which is cis-dominant. It can only affect the gene or the reading frame from which it is produced. Okay. And C for constitutive. Do you know what is the meaning of constitutive? Constitutive means the thing which can go on for so long. If there is a OC mutation, like without any condition, they will go on. Like for example, in this case, even in the presence of lactose or the absence of lactose, it will not bother. Okay, they will continue their work. That is constitutive. Like for example, there was some child which was so stubborn, they will not listen to their parents. They will do whatever they want. Like even if their parents scold or not scold, they will maintain their routine. They will maintain their bad habits of not studying. So they are very stubborn. So constitutive, you can relate with the stubbornness, or you want to do, you will continue that. You will not listen to anyone. So I'm coming to this letter. You have to first understand. So as because they are diploids, partial diploids, you can see two chromosomes here. Okay. So listen. So see very carefully. So in the first chromosome there is lacI plus O plus Z minus Y plus A plus, and in the F segment, so this is the chromosomal segment and this is the F dash segments. So which I have shown you. So this is the chromosomal segment, the first one, and this is the plasmid segment. Okay, this is known as the plasmid segment. So here in the chromosomal segment, these are the conformation of the gene, and in the F dash segment there is lacI plus, lacOC, point to be noted, there is a mutation, lacZ plus, Y minus, A plus. So what will happen? First of all, we have to check the conformation or the condition of the inhibitor gene. In both the case, you can see I plus lacI is the wild type, I plus. So you know that from I plus, the functional repressors will be made. Those can function very nicely. So from each of the chromosome, functional Lac repressors are made. And I told you, as Lac repressors are transdominant means that their product can travel and can affect another part. Here, these chromosomes Lac repressor can travel here. These chromosomes Lac repressor can travel there. Okay. So you can see they are traveling to each of them, and as because both of the chromosomes have lacI, they can bind to their chromosomes also, and they can travel and bind to the other part as well. So first of all, they will bind with the operator region. But what will be the case of OC? Point to be noted. Here the Lac repressor can bind to the O plus very easily because O plus is the wild type, and this Lac repressor can recognize the O plus very easily. But what will be the in case of OC? OC, the operator is mutated. As such, the Lac repressor can't recognize the operator and can't bind with the operator region. So here the Lac repressor cannot bind with the operator region. So you understood what happens? Even if the lacI is wild type, lacI product is coming from its own chromosome, lacI product is also coming from that chromosome, but the Lac repressor can't bind with the operator region because OC mutation is mutated operator. OC mutation produced mutated operator, and that operator region can't be recognized by the Lac repressor protein. So as a result, what will happen? Even in the absence of the inducer, that is lactose or allolactose, in this case, the second segment, that is the F dash segment, the plasmid segments Lac Operon will be turned on constitutively. So that is known as constitutive expression. So they will not bother even if lactose is present or lactose is absent. This operon, the F dash segments operon, will be turned on always constitutively because of the OC mutation present. Because of the OC mutation present, no Lac repressor can't bind with the operator. So the operator is free. Then the RNA polymerase can bind with the promoter region and can start the transcription of this segment. Okay. So we have to consider 2 points. Absence of lactose, presence of lactose. In the absence of lactose, I'm giving you absence of lactose and presence of lactose. You can see in the absence of lactose, the first chromosome can't be functional because the Lac repressor can bind to the operator region and can block the operator region, so RNA polymerase can't move. So from here, no transcription, you can see no transcription, no proteins made. So in the absence of lactose, the first chromosome, no expression. But the second chromosome, you can see, as the Lac repressor can't recognize the OC promoter, so there will be transcription and translation. Okay. There is transcription and translation, and I'm very happy. So in the absence of lactose, the second chromosome is turned on. Now the second condition is presence of lactose. In the presence of lactose, what will happen? Now for the first chromosome, if there is a presence of lactose, then the lactose can bind with this repressor region and can block the repressor. Then the repressor can't bind with the operator region. Then the operator will be free and the transcription will be on. And in case of second operon, there will be no need of lactose because the operator region is always free. The repressor can't bind with the operator region. So no matter, doesn't matter that if lactose is present or not, the second chromosome will always be on. This is known as constitutive expression. So in the presence of lactose, the first chromosome will be turned on and the second is always turned on. So this is the scenario. You have to understand these factors. This is very important. Okay, in any kind of problem, these type of problems will come in your exam. First you have to look at the lacI region, that what is the expression of the lacI. Then you have to decide that whether it is trans-dominant or cis-dominant. Always lacI is trans-dominant. And then you have to check the operator region, whether the operator region is wild type or mutated type. Then accordingly you have to decide. We will practice more, then it will be easy for you to understand. Okay, so this is the case. Now another thing from here I want you to know, that is that whenever you can see that lacY minus, even if this gene is this reading frame is constitutively active, if there is Y minus means constitutive non-functional permease. So Y minus means the Y minus the permease gene is mutated, so the product will be non-functional. Okay, or rest of the product, that is Z plus, that is constitutive beta galactosidase, A plus constitutive transacetylase. And why the constitutive word is written here, I hope you understood, because lacOC, because of the lacOC, because lacOC is there, that is why no repressor can recognize the operator and can't bind with the operator region. So operator is always free. RNA polymerase can move through the reading frame very easily. So in the absence of inducer also, the Lac operon is on. So that is why it is known as constitutive expression. Okay. Now this is the example when inducer is present. I have already told you, when inducer is present, you can see the allolactose is present. The allolactose can bind with the repressor. Now the repressors are inactive. The inactive repressors can't bind with the operator region. Then transcription is on. Beta galactosidase, permease and transacetylase made. Again I am telling you, Z minus means beta galactosidase will be produced, but it will be non-functional. Okay. And in this case, as there is lacOC, so no point of the inducer, so the inactive Lac repressor cannot bind and there is no need of the inducer molecule, so the expression will be turned on always. Okay. Now I will let you know the effects of lacI mutation in this region. Now here I will let you know about the mutation of lacI minus. I hope you remember that I told you lacI minus produces non-functional repressor. That means it can't function properly. It doesn't have that ability to switch off the operon. So you can see from the lacI minus, mutant repressors are made and mutant repressor can't block the operator region. Mutant inactive Lac repressor cannot bind to the operator, so the operator is free always. So the RNA polymerase can bind with the promoter region and can start the transcription, and as because transcription is on, so beta galactosidase, permease, transacetylase can be made. And why it is constitutive? Because it will not bother for the presence of inducer, that is presence of lactose or allolactose. I am giving you the point very easily that always constitutive means even if the inducer is absent, your Lac operon will be turned on. That is the main point. Okay, even if the inducer, that is lactose, is absent, your operon is turned on. In this case also you can see there is no need of the inducer, that is lactose, because I minus is already non-functional. If I minus is non-functional, there will be no point of Lac repressor to block the operator, so the operator is always free, so the Lac operon is always on. So we have studied also the lacI mutation. Okay, now we have to look at the partial diploid. The real problems will come in your exam with the partial diploid, with this diploid system, like chromosome 1, chromosome 2. There will be 2 chromosomes chromosome. Why I told you? Because this is the E. coli chromosomal segment and this is the F- segment, this is the plasmid segment. Okay, the real problem will come in your exam from this diploid system. So now here what we can see, first we have to look at lacI gene. Here in this case lacI is plus and in this case lacI is minus. What will happen, dear students? From lacI wild type repressors will be made, those are functional. Okay, and from lacI minus mutant inactive repressors will made, so that can't function properly. Now what is the idea? I told you that you have to always remember for lacI, that is your lacI is trans-dominant in nature. So even if this repressor is mutated, the operator region is blocked by the repressors made from chromosome 1. Okay, so here you can see chromosome 1 is making lac repressor that can block this operator, that can also come across and block the repressor and block the operator of number 2 chromosome as well. Okay, and this mutant, so mutant can't bind, you have to remember that this mutant can't bind. Okay, so that's sad. This chromosome could make its own operon on, but as because they are diploids, this lacI will interfere in this chromosome's expression as well, as because they are trans-dominant. So these chromosomes lacI plus is inhibiting the chromosome of this the operon of this chromosome as well. So the lacI plus from this chromosome is producing wild type lac repressor and it can inhibit its own operator as well as the operator of the chromosome 2. So what will happen, dear students, in the again we are going to that part, in the absence of lactose and the presence of lactose, absence of lactose and the presence of lactose. What will happen, dear students? In the absence of lactose, you can see the active repressors are made and they can bind to both of the operator. So if operators are blocked, there will be no point of turning on the Lacoperon. The Lacoperon is turned off in both of the chromosome in absence of lactose. But what will happen in the presence of lactose? I think you can relate. If lactose is present, then the lactose can bind with the repressor region and can block the repressor, then the operator will be free and transcription will be turned on. So for both of the chromosome, transcription will be turned on only in the presence of lactose. So here you can see in the presence of lactose, you can see the operators are, the lac repressors are blocked. So neither inactivated nor mutated repressors can bind to the operator. Inactive repressors so can't bind, that we know, but active repressors are also can't bind because active repressors are also deactivated by the inducer molecule, that is allolactose or lactose. Now the transcription can go on smoothly and beta galactosidase permease transacetylase is made. Again here also after the blocking of the repressor by the inducer molecules, beta galactosidase permease and transacetylase are made from another segment. Okay, so this is the overall scenario. Now you can say by this point that both of the synthesis is inducible synthesis. Okay, from this chromosome also it is inducible synthesis, from this chromosome also it is known as inducible synthesis. Inducible synthesis. Why they are known as inducible synthesis? Because in the absence of lactose, both the chromosomes are turned off. They need lactose to turn on their operon. They need induction to turn on their operon. That is why both of the chromosomes are inducible. But in this case, which I have shown you before, this is known as inducible synthesis. But in case of lacOC mutation, whenever you see lacOC mutation, that will be constitutive because they don't bother if lactose is present or absent. But here lacO plus is present, so in the absence of lactose it will be blocked. So in the for the first operon, only in the presence of lactose the operon can be turned on. So this operon is known as inducible expression, and the second operon is known as constitutive expression. I hope you understood, because constitutive means here is lacOC. Even if repressor is present, the lacOC can't be blocked by the repressor, so no need of inducer. So it is constitutive. But in this case, lacO plus is a wild type operator, and wild type operator can easily be blocked by the repressor. So when inducer is present, the repressors will be blocked, and the operon can be functional again. So that is inducible, that needs an induction. So okay, so this is another point. So in case of diploid system, you have to understand that which operon is inducible, which operon is constitutive. Okay. So we have completed some of the mutations, and we have also came across their examples as well. Now we will come across the example of lacIs, that is super repressor. Okay. I told you, I think you remember, that super repressors can't be blocked by the inducer. So let's see. For the first operon, that is lacI plus, lacO plus, Z plus, Y plus, A plus, all the wild type. For number 2, lacIs, O plus, Z plus, Y plus, A plus. So first of all, our first work is to see all of the genes and their condition. So for operon 1, lacI plus will produce wild type repressors. So you can see the wild type repressors. And the second operon will produce IS, which produce mutant repressor. In the presence of inducer, what will happen? That this inducer allolactose can block the repressors made by I plus very easily. But what it can't block is that super repressor. Even if you are giving the inducer molecule, the inducer molecule cannot inactivate the mutant super repressor. The mutant super repressors are so powerful, so active, that they can't be inactivated by the inducer molecule. Okay, they are super reactive. So that is why these mutant repressors can bind with both of the operons, because you know that any kind of inhibitors mutations are transdominant, so it can bind with its own breeding frame as well as to the reading frame of the operon 1, and can block the expression. So even in the absence of or the presence of the inducer, nothing will happen, because if in the absence of lactose only, the mutant repressor can bind with the operator, and even lactose is present, they can't inactivate the mutant repressor. Okay, so allolactose or inducer can inactivate the wild type I plus repressor, but can't inactivate the mutant repressor. So I'm writing it to you that inducer can inactivate or block the wild type, that is I plus repressors, but can't block the IS, that is super repressors. Okay, so that states clearly that in case of super repressors, the inducer binding sites are mutated. That is why the inducers can't bind, the inducers can't recognize these super repressors, and super repressor can bind with the operator region very easily, and no transcription is there. So this operon is known as uninducible. both of them, because they can't be induced. In the absence of lactose also, Lac repressors can bind, and in the presence of lactose also, because lactose can't induce the operon, because lactose can't bind with the super repressor. Okay, so both of the operons are uninducible. I hope you understood the every mutation, everything, students. In the next class, I will make you practice some of the examples of these operon system. Then you will get the idea very clearly. I'll request you to watch the video very clearly by pausing, by listening the points very, very clearly, and rewatch the video to get the best idea. I have tried my best to give you very lucid explanation. I hope you understood. If you have any questions, you can comment below and I will happy to answer you all. As I always say, fight for your dreams and your dreams will fight for you. Dear students, please study nicely and make your dreams come true. I'll meet you again in the next class with the problems of the operon system. All the best to all of you. Thank you.